The overall goals of this procedure are to demonstrate how the anti-inflammatory Aspirin-Fumarate Prodrug (GTC-PFE) is synthesized, and how it improves anti-NFκB and anti-cancer stem cell activity in breast cancer cells.
This method can help answer key questions in the cancer therapeutics field by showing how we can repurpose or even improve anti-inflammatory drugs to target therapy-resistant breast cancer stem cells. The main advantage of this technique is that we utilize the multidisciplinary approach to obtain and characterize the improved aspirin-fumarate prodrug. Demonstrating the synthesis of the aspirin-fumarate prodrug, GTC-PFE, will be Loruhama Delgado-Rivera, a graduate student in Dr. Gregory Thatcher's laboratory, whereas the anti-NFκB activity of GTC-PFE in breast cancer cells will be demonstrated by Gergana, Georgieva, a pharmacy student researcher in Dr. Jonna Fraser's laboratory.
Using a plastic plunger syringe, measure 0.81 milliliters of methanol and mix it in 10 milliliters of water in a round bottom flask. Cool the resulting mixture to 0 degrees Celsius by placing the flask in an ice water bath. Add 2.48 milligrams of 4-hydroxybenzyl alcohol to the reaction mixture. Stir until the solution is clear. Next, prepare a solution of O-acetylsalicylyl chloride in anhydrous toluene by dissolving the solute in the solvent in a separate flask. Using a plastic plunger syringe,